Chapter 77. Semen, Introduction. Semen is a white or gray fluid that contains sperms. It is the collection of fluids from testes, seminal vesicles, prostate gland, and bulbourethral glands. Semen is discharged during sexual act, and the process of discharge of semen is called ejaculation. Testes contribute sperms. Prostate secretion gives milky appearance to the semen. Secretions from seminal vesicles and bulbourethral glands provide mucoid consistency to semen. Nature of semen. At the time of ejaculation, human semen is liquid in nature. Immediately, it coagulates, and after some time it becomes liquid once again, secondary liquefaction. Fibrinogen secreted from the seminal vesicle is converted into a weak coagulum by the clotting enzyme secreted from prostate gland. Coagulum is liquefied after about 30 minutes, as it is lysed by fibrinolysin produced in prostate gland. When semen is ejaculated, the sperms are non-motile due to the viscosity of coagulum. When the coagulum dissolves, the sperms become motile. Properties of semen. Specific gravity, 1.028. Volume, 2 ml to 6 ml per ejaculation. Reaction, it is alkaline with a pH of 7.5. Alkalinity is due to the prostate fluid. Composition of semen. Semen contains 10% sperms and 90% of fluid part, which is called seminal plasma. Seminal plasma contains the products from seminal vesicle and prostate gland, Fig 77.1. It also has small amount of secretions from the mucous glands, particularly the bulbourethral glands. Sperm. Sperm is the male gamete, reproductive cell, developed in the testis. It is also called spermatozoon, plural equals spermatozoa. Matured sperm is 60 long. Sperm count. Total count of sperm is about 100 to 50 million/ml of semen. Sterility occurs when the sperm count falls below 20 million/ml. Though the sperms can be stored in male genital tract for longer periods, after ejaculation the survival time is only about 24 to 48 hours at a temperature equivalent to body temperature. Rate of motility of sperm in female genital tract is about 3 mm/minute. Sperms reach the fallopian tube in about 30 to 60 minutes after sexual intercourse. Uterine contractions during sexual act facilitate the movement of sperms. Structure of sperm. Sperm consists of 4 parts, Fig 77.2. Head, neck, body, tail. Head. Head of sperm is oval in shape, in front view, with a length of 3 to 5 and width of up to 3. Anterior portion of head is thin. Head is covered by a thin cell membrane, and it is formed by a condensed nucleus with a thin cytoplasm. Anterior 2/3 of the head is called acrosome or galea capitis. Acrosome. Acrosome is the thick cap-like anterior part of sperm head. It develops from Golgi apparatus, and it is made up of mucopolysaccharide and acid phosphatase. Acrosome also contains hyaluronidase and proteolytic enzymes, which are essential for the sperm to fertilize the ovum. Neck. Head is connected to the body by a short neck. Its anterior end is formed by thick disc-shaped anterior end knob, which is also called proximal centriole. Posterior end is formed by another similar structure known as posterior end knob. It gives rise to the axial filament of body. Often, the neck and body of sperm are together called midpiece. Body. Body is cylindrical with a length of 5 to 9 and the thickness of 1. The body of the sperm consists of a central core called axial filament, covered by thin cytoplasmic capsule. Axial filament starts from posterior end knob of the neck. It passes through the body and a perforated disc called end disc or end ring centriole. Finally, the axial filament reaches the tail as axial thread. In the body, the axial filament is surrounded by a closely wound spiral filament consisting of mitochondria. Tail. Tail of the sperm consists of 2 segments, chief or main piece. It is enclosed by cytoplasmic capsule and has an axial thread. It is 40 to 50 long. Terminal or end piece. It has only the axial filament. Products from seminal vesicles. Products of seminal vesicles are given in figure 77.1. Products from prostate gland. Products of prostate gland are given in figure 77.1. Semen analysis. Analysis of semen evaluates the qualities of semen, which is useful to investigate the infertility. Parameters of semen analysis: volume, reaction and pH, liquefaction, sperm count, morphology of sperm, motility of sperms, pus cells and RBCs, fructose level, qualities of semen required for fertility. Minimum required qualities of semen for fertility are: volume of semen per ejaculation must be at least 2 ml. Sperm count must be at least 20 million/ml. Number of sperms in each ejaculation must be at least 40 million. 75% of sperms per ejaculation must be alive. 50% of sperms must be motile. 30% of sperms must have normal shape and structure. Sperms with head defect must be less than 35%. Sperms with midpiece defect must be less than 20%. Sperms with tail defect must be less than 20%. Applied physiology. Azoospermia. Azoospermia is the condition characterized by lack of sperm in semen. It is a congenital disease. It is also caused by excess use of corticosteroids and androgens. Oligosuspermia. Oligosuspermia is the low sperm count with less than 20 million of sperm/ml of semen. Oligosuspermia causes infertility. Teratozoispermia. Teratozoispermia is the condition characterized by presence of sperms with abnormal morphology. It is also called Teratozoispermia. It occurs in Crohn's disease, Hodgkin disease, and celiac disease. The abnormal morphology of sperm results in infertility. Aspermia. Aspermia is the lack of semen. It occurs due to retrograde ejaculation. Retrograde ejaculation is the entrance of semen into urinary bladder instead of entering urethra. It is due to dysfunction of sphincter of the bladder, which is caused by prostatic surgery or excess use of drugs. Aspermia leads to infertility. Oligosuspermia. Oligosuspermia is a genetic disorder characterized by low volume of semen. Hematospermia. Hematospermia is the appearance of blood in sperm. It occurs due to infection of urethra or prostate. It is also common in congenital bleeding disorder.